One of the 8 faculties of the University of South Bohemia in České Budějovice is the Faculty of Science, which focuses primarily on biological, chemical, physical, mathematical and computer science fields. The faculty also evolves its research activities within these disciplines. A part of the Department of Molecular Biology at the Faculty of Science is also the laboratory of Drosophila model for human diseases. The head of the laboratory, Dr. Tomáš, who was engaged in the issue during his internships at the University of California in Irvine in the United States, here with his team uses a fruit fly, Drosophila, as a model for human pathologies. Most genes associated with human pathologies have their analogues in the fruit fly genome. As a fruit fly is a much simple organism and can be easily genetically modified, it may serve, in some cases, as a convenient model for the investigation of biomedically interesting issues.
Basically, we are combining different approaches using molecular biology, classical genetics, developmental biology. We're using molecular biology to clone the genes, to modify them, to introduce mutations into them. Then we usually transfer these modified genes into flies. And using classical genetic crossings, we get the modifications or the mutations into the right genetic combination, so we can do the experiments with them. Then finally, we usually visualize the effects of these modifications using microscopy, for example, to look at what are the effects on the development of certain tissues and similar things. Basically, this really long use of Drosophila as a model allows us to modify the genes in any way. We can turn on genes, we can turn off the genes of interest in any way. We can introduce mutations into them. We can even order now certain mutant lines from the big Drosophila stock collections. Any mutations we need from this circa 14,000 genes, we can obtain just by ordering them. And this relatively simple genetic manipulations in Drosophila allows us to study these effects in vivo in the context of the whole organisms, compared to just working with the cells in the culture, which lack the communication with the rest of the organism.
In his research work, Dr. Doležal builds on his mentor, Dr. Peter Bryant of the University of California in Irvine, who has been dealing with the issue of mutants in Drosophila for more than 20 years and has always believed to be able to use fruit flies as a model for cancer. He's managed to identify homologous genes in humans and has found out that one of them shows a noticeably high frequency of somatic mutations in a tumor issue of patients suffering from breast cancer.
We are using these approaches or this model to see or to study, for example, cancer in flies. I followed in this project work of my mentor from University of California in Irvine, Professor Peter Bryant, who started more than 20 years ago to collect different mutant lines in flies with the overgrown tissues, and he was hoping to model the cancer in flies. And he found many such mutants, and afterwards they actually found, based on this work, they found a very high rate of mutations in human cancer, in human breast cancer. So we used the advantage of this Drosophila model, just because the human gene, CK1 epsilon, and the Drosophila gene called disc overgrown, which are homologs, they are very similar. So basically we could easily recreate the human mutations in the Drosophila genome, in the Drosophila genetic information. So we introduced these human mutations into Drosophila, and we were checking the phenotypes of such mutants, and we found very striking overgrowth of the imaginal disc tissues, which are important for the development of adult structures like wings. So for example, one imaginal disc got really overgrown compared to the normal disc of the normal larva. So it kind of suggested that yes, these mutations are really cause of the cancer.
Currently, Dr. Doležal's team is examining the role of adenosine in the regulation of energy metabolism during the immune responses induced either by pathogens or by tissues damaged when genetic tools and tricks were used.
The last project, which keeps us quite busy in the lab, is related to my work from the PhD program, which studied very interesting small molecule which is called adenosine. I created a mutant with elevated levels of adenosine in Drosophila, and this mutant recently showed us that it could be related to energetic metabolism. So we decided to use again this Drosophila model to study this potential role of adenosine in the regulation of the energy. We are using different infection in Drosophila. For example, now we have in the lab this parasitic wasps, Leptopelina boulardii, which are injecting their eggs into the Drosophila larva. And that mounts very strong, robust immune response in Drosophila. And that's a perfect model for us because we can actually study the immune response in the larvae which are normal and also in the larvae which are mutant, for example, in the adenosine receptor. And thus it blocks the adenosine signaling. To really study the effects of adenosine, we need to use more infections. So that's why I started this collaboration with Dr. David Schneider from Stanford University in California, who is using Drosophila model to study different types of infections. And we are hoping that together we can see the effects of adenosine during different types of immune responses against, for example, Salmonella, Listeria, and such pathogens. And hopefully it will eventually clarify the situation. What is the role of the adenosine during the immune response, if it is regulating the energy, and how this regulation is important for the mounting the proper immune response.
Dr. Doležal is going to acquire necessary experience of infecting Drosophila with pathogenic bacteria during his internship at Stanford University in the United States, which he is leaving for within the 7th Framework Program Marie Curie Action.